Why evolution is true. The origin of species. In 1928, straight out of grad school, Ernst Mayr, a German zoologist, took a trip to New Guinea to look for different species. He collected 26 different species of bird and 38 different species of orchids. Nature is seen to be discontinuous. Plants and animals usually fall into one of many distinct groups. An example is presented when telling the difference between animals such as lions, panthers, and tigers. Each have distinguishing traits that help us tell them apart, but yet they are all cats. So how do you distinguish species? The morphological species concept states that individuals that resemble one group more than they resemble members of a different group should be considered a species. But this doesn't always hold true because some animals like the anglerfish look completely different when observed by gender. The female anglerfish is a lot bigger than the male anglerfish, so this concept doesn't hold true. Ernst Mayr helped define the modern synthesis of evolutionary theory, proposing the biological species concept. Ernst Mayr defined species as a group of interbreeding natural populations that are reproductively isolated from other such groups. So what is a reproductively isolated group? It's a group of organisms that don't mix genetic material through reproduction, such as spawning periods of some corals are timed differently so that sperm and egg can't mix. Also, pheromones or mating displays may be attractive to one species but not the other. So does this concept always work? No, because some organisms like the bacteria and some fungi don't follow these principles.
A hypothetical example would be a species of flowering plants that was split up into two sections by a mountain range. If one side of the mountain range is inhabited by a large group of hummingbirds but only a few bees, the flowers would become red. Red is a color that birds are attracted to, produce a lot of nectar which keeps the birds coming back, and have long tubes to accommodate the birds. On the other side of the mountain, where you would find few hummingbirds and a lot of birds, the flowers may become pink to attract bees as pollinators, shallow nectar tubes with less nectar, and have flatter flowers to accommodate the bees that want to land on the plants to collect the nectar. Eventually, the two populations would diverge and would specialize in pollination by a single type of an animal. This would result in them being reproductively isolated because each type of flower would be pollinated by a different type of pollinator. Different mutations affecting male behaviors could appear in different places. Let's say longer tail feathers versus orange color in one population. Eventually, females of one population would prefer males with long tail feathers, and the females in the other population would prefer males with orange feathers. This allows for the species to even have more differing mating preferences and would prevent them from mixing genes, and later be considered different species. The virility and viability of hybrids are accidents of genetic divergence which is caused by natural selection or genetic drift. When two populations that evolve along different pathways for long periods of time can have genomes that can change enough that when they're put back together, they don't work well together. This will interrupt development, which can lead to premature death or infertility. Theory of geographic speciation, which states that the evolution of genetic isolation between populations requires that they be geographically isolated, is important because it doesn't enable the species to interbreed. For example, insects inhabiting an area with two types of plants, with each plant having different toxins, nutrients, and odors. As insects begin to adapt to one plant, they also mate with insects adapting to other plants. This constant mixing of genes will keep the gene pool from splitting into two species. Evidence of geographic speciation, which include fossil records, embryology, are a few examples of data that are evident. Speciation is a slow process unless it is happening bacteria. Studies conducted by Coyne and Allen showed evidence that starting with one ancestor, it takes roughly between 100,000 and 5 million years to evolve two reproductively isolated descendants. Because of this, it is impossible to witness the process in one lifetime. Because the events are hard to observe, predictions are made from the theory of geographic speciation. The first theory that can be tested is if speciation depended on geographical isolation, then there must have been ample time for opportunities for population to experience isolation. There are many forms of geographical isolation, like mountain ranges rising, deserts forming, continents drifting, and droughts dividing forests into grasslands. All these events could lead to species dividing. However, these could not be the only reason why. Accidental long-distance dispersal by birds, there has been ample time for species to split their ancestral branches because this event occurs exponentially. Reproductive barriers are tested in the laboratory, where scientists force animals or plants to adapt through evolution to different environments. Second prediction involves the geography of an area. If populations have to be physically isolated from one another for new species to form, we should find new species in different nearby areas. By looking at the DNA sequences of the species, you can get an estimate of the time in which they split. After narrowing down the time lapse difference, you can look at sister species and look for differences. A great example would be the snapping shrimp, which are isolated by the Isthmus of Panama on each side. As the isthmus rose, the shrimp were divided up. This can also happen with plants. Botanists believe that each of the 9 pairs used to be a single species that were divided up due to climate change, which wiped out the forest in between. There are examples of geographic speciation still occurring today in nature. A great example is the orchid Satyrium halaki, which lives in South Africa. To attract pollinators, the orchid has evolved long nectar tubes in its flowers, and pollination can only occur when the long-tongued moths or flies get close enough to collect nectar. However, in the coastal regions, there are only short-tongued bees, so the flower has much shorter nectar tubes. If all were to share a location, there would be some problems because short-tongued pollinators wouldn't be able to pollinate flowers with long tubes, and vice versa. The final prediction that can be made is that populations which are physically isolated will get an increase in reproductive isolation. A great example is a study that was done on the Drosophila species of flies, which has a pair of common ancestors that has diverged at various times in the past. The study measured 3 types of reproductive barriers, which included mating description, sterility, and viability of hybrids, and the results yielded that the reproductive isolation between species increased steadily over time. Results of speciation shows that isolated populations are populations that show no reproductive isolation, increasing degrees of reproductive isolation, and complete isolation. In conclusion, isolated populations diverge, and when the divergence has gone on for a sufficiently long time, reproductive barriers develop as a byproduct of evolution.
Creationists do not believe in evolution. Their number one claim is always the same. If it's not possible to see new species evolve during our lifetime, then speciation does not occur. But that's insane because most of evolution happens, but we can't see it in human lifetime. It takes too long. But an example is the life cycle of the star. According to them, stars do not evolve because we cannot see it, but we can trace back. Another example is language. Languages do not arise because we cannot see it evolve, but we can trace that as well. Thanks to historical reconstruction, we can actually make valid arguments for these processes. Now we move on to geographic isolation of populations. We know that this is the most common way of speciation to take place. Closely related species that live in the same area could have actually diverged from each other in an earlier time when their ancestors were geographically isolated. But it is argued that new species can arise without a need for any geographic separation or barriers. This is what's known as sympatric speciation. Sympatric speciation happens when we get a new species without geographic barriers. The only problem with this is that it's hard to split one gene pool into 2 while its members remain in the same area. The reason for this is because there's constant interbreeding between these diverging forms. Interbreeding will always be pulling them back into a single species. But this is possible under restrictive conditions that may be uncommon in nature. Find evidence for sympatric speciation. Looking for evidence is hard because related species living in the same area could have been because of climate or habitat changes that have occurred in the past. They could have come into contact at another place at another time. But we can find evidence in small parts of isolated waters and land. The key factor in this, though, is that the isolated areas have to be so small that they could not possibly contain any geographic barriers. An example of sympatric speciation is the cichlid fish. They live in 2 tiny isolated African lakes in Cameroon. Each lake contains a different mineralization of species. Each recently descended from a common ancestor. One lake has 11 species and the second one has 9. Although it is not known exactly why and how this happened. A second example are palm trees. On Lord Howe Oceanic Islands in the Tasman Sea. The island is small and it contains 2 native species of palms, the Kentia palms and the curly palms. They have a common ancestor that lived on the island in the past. Surprisingly, though, sympatric speciation does not occur when it has the opportunity to happen. There are many habitat islands that contain a good number of species, but none are their closest relatives. Trevor Pierce examined bird species in isolated oceanic islands looking for close relatives, and they could not find any. Out of 46 islands, not one contained endemic bird species that were each other's close relatives. Similar results were found for the Anolis lizard. Nothing was found. Nothing. Close related species are not found on islands smaller than Jamaica. There are 2 special forms of sympatric speciation that are common in plants and are the only cases of speciation in action. These species are actually forming during a human lifetime, which means that it's speciation happening right before our eyes. One is called allopolyploid speciation, and the second is autopolyploidy speciation. Let's examine this. Polyploid speciation does not start with isolated populations of the same species. Instead, it starts with a hybridization of 2 different species that live in the same area. For this, though, it is required that the different species also have different numbers of chromosomes. Therefore, a hybrid's chromosomes do not pair up when it tries to make pollen or ovules, and so it is sterile. If the chromosome could be doubled, the chromosomes could have a pair and the hybrid could become fertile. And it will give us a new species. The second type is autopolyploidy speciation. This polyploid speciation does not always require a hybridization. A polyploid can arise just by doubling all of the chromosomes of a single species in what is called autopolyploid. And this can also give us a new species. Autopolyploid mating with other polyploids can produce a fertile hybrid. Autopolyploid mating with the original parent species produce sterile hybrid. How do we get polyploid speciation? A rare event has to occur in 2 successive generations. The formation and union of sperm and eggs with abnormally high numbers of chromosomes. As weird as this sounds, this is not rare, at least in plants, since they can produce millions of eggs and pollen grains. It is estimated that as many as 1/4 of all the species flowering plants were found via polyploidy. Polyploid species are found in nearly all groups of plants, except trees. Polyploid plants. Many plants used for food or decoration are polyploids, or sterile hybrids that had a polyploid parent. 2 examples from our very own kitchen. One is wheat, that have 6 set of chromosomes that arose from complicated crosses that involve 3 different species. Commercial bananas are sterile hybrids between 2 wild species having 2 sets of chromosomes. from one species and one set from another. Fun fact: the black specks in the middle of the banana are absorbed plant ovules that do not become seeds because their chromosomes cannot pair properly. Polyploids in animals is more rare, but sometimes it happens in fish, plants, worms, and reptiles. Most of these reproduce asexually. There is one sexually reproducing polyploid mammal, the red viscacha rat of Argentina. It has 112 chromosomes, which is the most seen in any animal. It is not known why polyploids are so rare, but maybe because it disrupts with the mechanism of the X and Y sex determination, or with the inability to self-reproduce. Polyploid speciation is different because it involves changes in chromosome numbers rather than changes in the genes themselves. Also, it is faster than normal geographic isolation because a new polyploid species can arise in 2 generations. This gives us the chance to see speciation happen in real time for those who want proof of speciation in our lifetime. There are at least 5 plants that arise this way. One is the Welsh groundsel, and we will look into that. The Welsh groundsel. This is a flowering plant in the daisy family, first seen in North Wales in 1958. This plant is a polyploid hybrid between 2 other species. One is the common groundsel and the other is the Oxford ragwort. But now let's look at the evidence that proves that this indeed is a hybrid. Evidence that this is a hybrid. For one, it looks like a hybrid because it has features from both the common groundsel and the Oxford ragwort. Second, it has the chromosome number 60, which is predicted because one parent has 40 and the other has 20 chromosomes. Genetic studies show that genes and chromosomes of the hybrid are combinations of the parental species. And finally, it was artificially produced in the lab and looks exactly like the one seen in the wild.